see if I can find it. File. Maybe I shouldn't do it right now because I don't want to open any of these files that I don't necessarily want you guys to see. All right, so I will post right here in Module 2, right, the file, and it has the ones that weren't on the test marked on it, okay? So you don't have to study those. You have to study all the ones that were on the test for the final, right? As it relates to Module 3, I'm going to add under the lectures all the PowerPoints, and this is the, I had posted this once before for you guys. This is the outline. Here, I want to just view it. But again, these are the objectives right from your book. So for the test that I'm going to give you guys a take-home test, I'm going to give you a sheet with all the objectives numbered. Right? They call them learning outcomes here. Right? So here it is highlighted, learning outcome. And they're going to be numbered, like 1 through whatever it is. And you need to match that number with the question on the test. Right? So I know you can match up objectives with test questions. Right? Make it a little bit more challenging for you guys. So then the other question is connect assignments. Right? So for these chapters, I haven't done connect assignments yet. But I think it would be good for me to do them for you guys, because after you hand in the test, because again, you're going to be looking at your notes, right? There's no way I can stop you, so I expect that, right? You're going to be looking up this stuff on your notes. Putting connect questions so that way when you guys are going to study this material for your final, right, which isn't going to be open notes, you can kind of quiz yourself on those connect assignments before the final exam, right? So I will post some connect assignments for you guys to work on, but I don't expect you to do them until after you hand in this test as you're studying for the final. But know too, those old ones that were on there before, you can redo those as you're studying for the final as well, right, to help you practice and see if you remember the material. Does that make sense? Okay. So Sherry and Ashley came in late. Remind me after because I already started recording. You came in late too, that's right. A bunch of you guys. Okay. Any questions, problems, concerns regarding that? Okay. So chapter 12 we started, right? Genetics. Module 3, chapter 1. Chapter 12 we started, and that was before we even took the test on 2, right? So that feels like ancient history, doesn't it? So open that one up. So this is 1. Right, okay. I don't know if I finished 2. Oh, this is all the slides. I didn't even make it back to it. Okay. Hence why I didn't post it. Okay, we're going to do a quick flash through. So we talked about a gene and we talked about the transcript. And where's the slide that I want to get to? We talked about structural genes actually encode proteins. Right, the steps in the process go in the central dogma. We go from DNA to RNA to the polypeptide that forms the protein. And we defined, right, at this point you should definitely know what transcription versus translation means. And we talked about these different regions within the DNA, right, that control whether the transcript is actually made, right? They may have a regulatory sequence, a promoter where the RNA polymerase binds. So these are definitely components that you need to know, right? The ribosome binding site for the messenger RNA and the codons. And I want to get into this today, which is going from messenger RNA to translation, which is actually in the next chapter. I'm going to skip over, we talked about the different RNAs, about the promoter and the particular sequence and why it's got a lot of A's and T's, right? Because that's less hydrogen binding and you can open that up. How the enzyme actually attaches. Part of it actually is interacting with that sequence in the major grooves. And the sigma factor does that. And how only one strand is used as a template, right? So sometimes it's usually it's either the top or the bottom strand, depending on how the gene is oriented within the chromosome. So that was last time. So that ended we talk about elongation, spliceosomes, yep, the promoter, the enzyme. It's right there. Oh, look, ended. See, I left myself a note. All right. We don't need all that in this one. We can delete that and save. Okay. So the mechanisms vary between bacteria on how they stop, and I think this is pretty straightforward stuff. So like I said, I wanna skip over that for now, and let's see, it goes into processing, yeah, RNA modification for eukaryotes. Okay, so they talk about some of the experiments that were done, like this one, to figure out that eukaryotes actually, I'm looking for a particular slide. Sorry. Probably like making you guys feel like you're going dizzy, huh? Okay. I guess it's this one. I hate how tiny this is. We will make it bigger. Cut it in half. I hate when they put stuff in tables sometimes. Alright, that should be good. Okay, that should be somewhat readable. The picture is what I care most about. You guys can read the stuff in your book. So for eukaryotes, and I still haven't found that picture for you guys, bacteria will make a big, long transcript of messenger RNA, right? And remember, they don't have a nucleus, so ribosomes will even start attaching to that messenger RNA at the ribosome attachment sites and start making proteins, the polypeptides, right from the messenger RNA. Where in eukaryotes, right, we make the messenger RNA, and for the most part it travels out of the nucleus, and those proteins are going to be made in the cytoplasm with the ribosomes there, or maybe attached to the rough endoplasmic reticulum. One of the things that they found out as they were investigating this was that the messenger RNA actually gets processed before it is translated by the ribosomes. Okay, and there are several different things that can happen to it. So in this case, the processing actually removes some of the ends of the messenger RNAs, right? So this is the original transcript, right? And pieces are cut off and then they're degraded, and this is what actually is read by the ribosome. Why these things happen, right? We always ask why, don't we? There's some speculation as to why, but I don't want to necessarily get into that today. The really interesting thing, okay, so what the ends are cut off and degraded, is when they noticed that there was this phenomenon where there were segments in the middle that were removed and the two pieces were spliced together. So of course they called that splicing. Do any of you guys know what this segment that they removed in the processing is called in the DNA code? They're called introns, for intervening sequences. They don't actually get used to make the protein, right? Because the messenger RNA is processed so much so that that section is spliced out. The exons are actually what are put together and are actually used by the ribosome to make the protein. I was like, it's kind of bizarre, huh? Why? It seems kind of wasteful, doesn't it?
I would say it seems more efficient because it takes up the part that you don't need.
Yeah, but why even have that part you don't need?
That's what makes DNA so complicated.
It makes it so complicated. Yeah, it's definitely what makes it very complicated, especially when we're talking about eukaryotes. So the other thing that is somewhat to our benefit is that, and again, this probably has to do with the fact that this message really does travel. Unlike in bacteria, right, where it can be right there being made off of the DNA and already be utilized by the ribosomes. Messenger RNA for eukaryotes travels out of the nucleus. So it makes sense for certain modifications that happen. Right, so one of the things that happens is they attach the 7-methylglucosamine cap. This is referred to as the 5' cap because they put it on the 5' end of the messenger RNA. And as I said, it plays a role in getting it ready to be spliced, but also for its kind of its exit signal, like you're okay, you can leave the nucleus now, right? Here's your hall pass, you can leave now, right? And go where you need to go. And so this is important, they found, for binding of the ribosome. If that cap isn't there, the ribosome won't bind, right? For eukaryotes, right? So this is kind of a signaling method. So the other thing that happens is there's an attachment of a string of adenine-containing nucleotides. So they add a whole bunch of A's to the end. So again, this is at the other end, at the 3' end, and so that's why again it's given the 3' name. But it's called poly-A tailing. Again, it's the tail, it's the end, right? So remember, we go 5' to 3'. All the enzymes read in that direction. Hopefully we've got that down, right? So it gives them a little tail and to the end. And again, they found that this is important. If it doesn't occur, then translation can't occur. It sometimes happens on bacterial RNAs. But notice that the capping only happens on eukaryotes. So this is an important thing too, like when we talk about our immune system and when bacteria get into our body and their DNA differs, their messenger RNA differs, or viruses, their messenger RNA differs. One of the ways it differs is it doesn't have that capping, right? And so one of the ways our system recognizes that's foreign messenger RNA is the fact that it doesn't have the cap. Or you could say, you know, it also why it will be attacked, right, by our immune system. But some of them, of course, have gotten around that and it can get the system to work. The change of a base sequence of RNA after it has been transcribed. So remember that there are only U's in RNA, there aren't any T's. And the other modification that happens is that there is a modification that happens to some of the bases, right? Where one of the modifications we looked at before is like adding methyl groups. And so this does happen in prokaryotes. But eukaryotes, they methylate usually the cysteines specifically. So non-structural genes. So first of all, what is a structural gene? What do we define a structural gene as? One that codes for what? No, DNA is a gene, right? The genes are within the DNA. So if we call that segment of the DNA a structural gene, what does that mean? It goes from DNA to RNA, and what are we going to make? We're going to make a protein. Is all DNA, does it all get transcribed into proteins? No, it's not all structural genes, right? Some of it is to actually make RNAs, right? Remember all those special RNAs that we talked about before? Transfer RNAs, ribosomal RNAs that are part of the ribosome, right? The piece that goes in telomerase, right? That RNA that goes in the piece of telomerase. So they'll actually make a long transcript and then break those into their individual pieces. So they don't transcribe them as single sets of genes. It's almost like polytrinsic, like what bacteria do. And it makes sense for the ribosome because, again, you need several pieces to make it. So for the ribosome, it actually has 3 pieces of RNA that complex with the proteins to form the whole ribosome structure. And so when they transcribe this, they'll transcribe this whole piece of messenger RNA, of RNA. It's not a message. It's not going to be translated. It's RNA. It's ribosomal RNA at that, hence the little R. And then the enzymes will cleave it into its 3 distinct pieces that go into the ribosome. So this was discovered pretty early on, that it just had one promoter, right, and made one long transcript, and then it broke the transcript into pieces. And this happens in a region that forms within the nucleus referred to as the nucleolus. It gets really dense with all this RNA, right, that's being processed because you need a lot of ribosomes for a cell. Right? So they produce a lot. It's not membrane bound, right? And so now here's the functional pieces that will go into the ribosome. So transfer RNAs are also made as very long precursor RNAs. And both of them are cleaved into the individual ones. So exonucleases cleave the covalent bonds between the 2 nucleotides at the end of the strand, and endonucleases cleave the bonds within the strand. So when we look at the trimming, right? So here's the big long transcript, and it forms a two-dimensional structure. You have inner strand binding. Remember, this is a single strand though, right? You see, you can follow it along. It has a beginning and an end. So the exonucleases, right, cleave here off the end, and the endonucleases cleaves off right here. And others will cut other sections within. This region right here we're going to talk about next time is the, well, we might get to it today, is the anticodon. So remember, this is a sequence, right? Age, what are they showing here? Because I'm confused because they have some T's in here, and RNA is not supposed to have T's. Oh, it's a modified. It's been modified. Okay. Okay. So his experiment, he actually took, I want to get to that picture here. So he took what's called coding strand DNA. And so this region right here is complementary to the messenger RNA. So they can interstrand bind. So he took the DNA, he heated it up so the strands would separate, and then cooled it down. But when he separated the strands, then he added in the mature messenger RNA. And then he cooled it down. And so the messenger RNA bound to the complementary, right? The coding strand, remember, is the same sequence as the messenger RNA. Those were terms that we learned before the test. And then, so it can interstrand bind with its template, right? Because they're opposite to each other, right? So what he found was that when that happened sometimes, right, you would expect it to perfectly match. What happened, and it was pretty amazing what they did. They coded the DNA in such a way, and they were able to take pictures of it under electron microscope, right, because it's such large molecules. And so he saw these loops that formed, right, that the DNA would be looping out, and it wasn't interacting and covalently, or hydrogen bonding, I should say, with the messenger RNA. So what happened to this DNA? Why is it not in the messenger RNA? Right? Why is it looping out? Well, because that portion was cut out. And as I said, that's what we call the introns. So for the particular gene that he looked at, it had just one intron, one intervening sequence. So you can see here, right, under the microscope, there's that intron loop. Here's where the DNA is buckled out. That's that R loop. And that's because the messenger RNA is binding with it here to create a double-stranded structure, and also here on the other side. Right? And then this is the DNA, the coding strand, which is the same as the messenger RNA, sticking out. It's kind of amazing how they thought to do that, you know. Nowadays, do you think they do stuff like this? How do they figure out what's introns and what's exons and what's actually being used by the messenger RNA? What can we do nowadays that they couldn't do back then? All right, so this was done, right, in the 70s, I think. What'd you say, Angela? We can sequence DNA and RNA now, right? We can sequence it, right? Using machinery and stuff. Now we can get the actual sequence for the DNA, right? Get the sequence for the messenger RNA, and then we can match up and see what's actually in the DNA as opposed to what is actually in the messenger RNA. And you will see these segments, right, that don't end up in the messenger RNA. that are in the DNA code, those intervening sequences. So there's 3 different splicing mechanisms that have been identified. So there's the group 1 intron splicing and the group 2 intron splicing. And then the spliceosome, which uses a set of enzymes that work in conjunction. All 3 of them involve removing introns out of the RNA. The linkage of the exons in the RNA together, right? If we're going to cut pieces out, we've got to link them back together, right? So they all have to link those pieces back together if they cut out introns. So group 1 and group 2 intron are termed self-splicing. It doesn't require the aid of enzymes. So instead, the RNA itself functions as its own ribozyme, right? Its own enzyme. The difference between the 2 groups is the way in which the intron is removed and the exons are reconnected. It's pretty cool stuff. So they can get it to occur outside the body, right, in vitro, without additional proteins, although there are proteins that are needed for it to run in vivo. And they refer to these as maturases because they help enable. So they don't have enzymatic activity themselves, but they help in orienting the molecules so that the splicing can happen. So what they're showing you here is this guanine here binds back with part of the messenger RNA here, and this creates that loop, and that loop allows it to be removed, but it doesn't get removed until this segment gets attached back to the other part that's also the exon. In this other one, the loop forms off to the side. Do you notice it's off to the side, it's not within here, and then it attaches here. And so you have this loop right here, and it's the adenine here that's facilitating that loop structure that forms. So guanine here, right, the loop forms, and it's cut out guanine to guanine. In a spliceosome, it's actually proteins that have enzymatic ability that actually allow the looping out of this and then the adding back of this segment here. So before it gets spliced, it's called pre-messenger RNA, of course, right? And then afterwards, it's referred to as mature. The other modifications, like the capping and stuff like that, have to happen before it leaves the nucleus. So this is very common, the use of the spliceosome to cut out that intervening segment. So for the rRNAs, they're self-splicing in some eukaryotes. Found in a few protein-coding messenger RNAs, but also happens for the transfer RNAs, the ribosomal RNAs, within the mitochondrial DNA for fungi and plants, and in the chloroplast DNA. Rarely is that found for the transfer RNAs in bacteria. So the group 2, this is done for transfer RNAs where it's self-spiking using the group 2 mechanism. And ribosomal RNAs within the mitochondria for both fungi and plants and in the chloroplasts. And again, rarely is this done because, again, this isn't something that is very common for bacteria, which is much more common for eukaryotes to do the splicing. So obviously some RNAs are even involved in that process. And you can see the code. There are particular sequences within the intron and the exon barriers that help facilitate the splicing that happens. So here, those RNAs are recognizing and binding, right? And then it brings it together in the spliceosome, creating that buckle, right? And they'll cut the end off and then splice these two ends back together. Others will form that loop. So what's the advantage? You don't always have to splice it the same way, right? You don't have to put particular sequences together. Like exon 1 doesn't have to go on with exon 2. You can even cut out exons and put different exons together. And so they found that this happens, can be spliced out in different ways and actually generates different proteins. I'm trying to remember if we had a good picture of this. So a single gene can make 2 different proteins. They don't give us an example, huh, until chapter 16. Damn it. All right. We already mentioned the capping and adding the tails. All right, I want to get to some other stuff. We'll be very broad in that area. I want to go to 13. So this one's really raw. And of course there were cool guys that figured this out. There it is. That's where I want to be. No. No more memorizing scientists' names. We're good. We're done with that. I will promise you that, right? Just the ones we've already covered. That was enough, right? You guys are like, I have my list. Please don't add to it, Miss Erica. Right? Those are the important ones. But, you know, definitely read over. Oh, no, there's one other one. One last one. The very last one that we'll cover on Thursday. Barbara McClintock. She's the last one. One more girl to the mix. So this is our genetic code. They have pretty much worked it out, which is pretty amazing. And of course, they talk about the scientists that put this together and how they came about it. So we've mentioned this already because we very generally at the beginning of the other chapter, we talked about how we go from DNA to messenger RNA to protein, basically polypeptides. And most of us already know that the ribosome facilitates that. But now we'll also have mentioned these transfer RNAs. And we're going to look again at them. So once the messenger RNA is processed in the case of eukaryotes, right, and it's ready to be read by the ribosome, there's a particular sequence in that messenger RNA, again, the ribosome will bind to. And the ribosome itself contains RNA, right, so it helps recognize these sequences. And it's going to attach. But it's not going to start bringing in transfer RNAs that match up with an amino acid attached to it until they get to the very first codon, which is referred to as the start codon. So there's only one of these, right, and is AUG. And it codes for an amino acid. So that transfer RNA comes in, and that transfer RNA, the sequence on the end that interacts with the messenger RNA via the ribosome, is referred to as the anticodon. As you saw in that picture, I said, that's the anticodon. Anti meaning opposite, right? So what would be the opposite of this in RNA? So what would bind with an A? We're saying RNA, though. It's got to be U, right? And with the U, it's got to be an A, right? So we switch the U's out for T's, remember, when it comes to RNA. And then the last one is C, right? C and G is the same as it is in DNA and RNA. So that's what that codon, that anticodon would say in the transfer RNA. At the end of that transfer RNA molecule would be the amino acid. I probably should pull up a picture for us, huh? Pictures are better. Trying to see. All right, so there's one. There's a transfer RNA. So there's its anticodon, right? And there's some modified nucleotide, so that's why, this one is in a letter you recognize. But there's G and C. Let's see if they tell us. So this segment that sticks out is actually what binds to the amino acid at this end, right? So these are modified bases, right, are shown in blue here. There's some modifications that they do to the bases that are found in transfer RNA. So we're not going to get into the particulars of that in this class. But that's what they're marking there. So there's the anticodon, and it's kind of funny. They end up like this almost T structure, but the T actually stands for transfer, right? So they're going to transfer these into the messenger RNA ribosome complex, and this matches up with the messenger RNA and carries a particular amino acid based on the sequence of this transfer RNA. So here it is in action, actually. Here's a good picture of it. So you can see, right, this is the messenger RNA, there's the transfer RNA, and here is the amino acid that it's carrying. So it actually, enters into that complex. I'm thinking, yeah, right, so we're matching up. And it's read in groups of 3 nucleotides referred to as a codon, right? So you have your start codon, you have your sense codons, which actually code for amino acids, so the amino acids are being put there. And the ribosome is going to form that bond between the 2 amino acids, and that's referred to as a peptide bond. That's why when you make the big long string of amino acids, it's called a polypeptide, right? And for some proteins, that's the protein, right? It's going to fold back on itself, right? It's going to form beta sheets and alpha helices and form this globular 3-dimensional structure, right? Some proteins are actually made of many polypeptides, right? So RNA polymerase, DNA polymerase, right? We've talked about how it's made up of more than one protein or more than one subunit of that protein. Sometimes scientists get real particular on what they call a protein versus a polypeptide, right? Others of us badly, I guess, use them very interchangeably, those terms, but they are distinct from one another. Most scientists, when they say protein, they're talking about something that's functionally active and has all its pieces, that it needs to do its job. So I want to go back to the code. So how many amino acids do we have? Do you guys know? Naturally occurring, the most common is 20. So we have 20 different amino acids. But when you look at this code, right, if we read in groups of 3 nucleotides, and there's a possibility of 4 different nucleotides at each position, you end up with this table that we have here of 64 different combinations. And yet we only have how many naturally occurring normal amino acids? 20. So we have more code than we need, but if we only did 2, we wouldn't have enough, right? So we've got to have this 64. So there, it's said to be redundant, right? We have more code than we have amino acids. So guess what? For each amino acid, there's more than one code, right? And then they've found in the research too that as you saw with that one, some of them, that third position is modified because it isn't as important as the first 2 positions for certain amino acids. So let's look at a couple of them. And again, by no means do I expect you guys to memorize a table like this, but understand how it's used, right? So the very first code in the messenger RNA would be AUG, right? So there's that code. The transfer RNA would transfer in, and that would be carrying methionine. That's the amino acid. And amino acids are also abbreviated by 3-letter abbreviations. And the nice thing I like about this table is it actually has both the full name of the amino acid and the 3-letter abbreviation that's commonly used. This isn't biochemistry class either. We're not memorizing all these amino acids and their structures. Aren't you glad? Right. But note that 3 of these guys do not code for amino acids. They're what we call stop codons, or sometimes non-sense codons, right? I used this term last time. Sense refers to actually coding for an amino acid. So all the rest of them are called sense codons. S-E-N-S-E, right? Sense. These are non-sense or stop codons. And for the most part, for most organisms, there's 3 of them. That when the ribosome gets to this, no transfer RNA, right? It's going to end translation, excuse me. We're on to translation. So if you look at especially proline right here in this block, do you see how it doesn't matter out of the 4 nucleotides possible, it doesn't matter what's in this 3rd position? Proline's going to get put there as long as the first 2 nucleotides, right, are CC in the code. So what does the anti-codon on the transfer RNA have? To what? If this is C's in the messenger RNA, it's what in the transfer RNA? 2 G's. 2 G's. Yeah. Where for some of these it does matter, right? So notice right here, histidine versus guanine. If it's C, A, U, or C, it's going to be histidine. If it's C, A, or G, it's going to be guanine that's put there. So the ribosome, right, brings in the transfer RNAs that match up with the code, right, via their anticodon. They're carrying their specific amino acid. And the ribosome is going to ligate together the bonds, the peptide bond between those amino acids. So that strand of amino acids just keeps forming out one end of the ribosome. Transfer RNAs are transferring in and popping out as it's reading that message, right? So I know that for translation, they do have a very good animation. Okay. Did it open? It's opening. So I'll post this for you guys, but it's in your book, right? You can link to it in your book. So there's actually an enzyme that adds the amino acids to the transfer RNAs. We're not going to bother watching that. We're going to watch I know this one's good. I'm not sure about this one. Here, we'll play them both. See which one we like better, right? We'll vote. Okay. Let me make sure my volume's on. Let's see.
Translation is initiated by formation of an initiation complex consisting of the smaller ribosomal subunit, the first amino acid tRNA, and messenger RNA. The larger ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids can bind. One is called the peptide or P site, and the other is called the acceptor or A site. The initiating transfer RNA carrying its amino acid binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The amino acid carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site. The ribosome now advances a distance of one codon, and the first transfer RNA is released. A transfer RNA carrying the next amino acid now moves into the A site, and the ribosome shifts down by a distance of one codon. This is followed by the two amino acids on the transfer RNA in the P site being transferred to the third amino acid. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when the ribosome reaches a codon that does not code for an amino acid, called a stop codon. The ribosome dissociates into the smaller and larger subunits, and the messenger RNA and protein are released.
Pretty cool. All right, but like I said, did you see how those codes were matching up, right, for those? So I had watched this one before. I don't know if this one's any better. We'll see.
Chain elongation begins with the binding of a tRNA, which recognizes the next codon in the mRNA to the A site of the ribosome. This is catalyzed by the EFTU transcription factor and requires the hydrolysis of a GTP.
Oh yeah, I already like the new one, the first one better, huh?
The polypeptide chain is moved from the tRNA in the P site to the amino acid attached to the tRNA in the A site. Peptidyltransferase, a protein RNA complex present in the 50S ribosomal subunit, catalyzes the formation of this new peptide bond between the amino acids. The ribosome then translocates to the next codon. This process is promoted by elongation factor G and requires another GTP. This places the empty tRNA molecule in the E site of the ribosome and moves the tRNA containing the growing polypeptide chain in the P site. The next codon in the mRNA chain is positioned in the A site. The uncharged or empty tRNA in the E site then leaves the ribosome and a cycle of chain elongation is completed. Through subsequent cycles of chain elongation, the polypeptide chain continues to elongate one amino acid at a time.
Yeah, I like the first one better. I don't know, that was just me. She talks about some of the other particulars in this one, right? That gets you kind of lost. But you can still see the movement, right? And how these come in. That's really what I wanted to show you guys in this, right? The different components and how they come together to achieve this process of translating that messenger RNA in groups of 3 nucleotides known as codons, right? And so as I said and showed you guys, we know this, we've broken the code, right? The Morse code for figuring out what amino acid will be put there based on the codon in the messenger RNA, right? what scientists really are still struggling with is how does it go from that polypeptide, that big long chain of amino acids, how does it know to fold the way that it does to function the way that it does? That's still very much a mystery for us. So I think I've bored you guys enough for right now. I wanted to go over that general process today. So I am going to skip to 15, which is...
Skip 13 and go to 15.
No, that was 13. That was 13. That was 13. I'll post the whole thing for you guys. We're going to skip to 15. I'm trying to remember. Yeah, 15 is complicated regulation. Yep. So read over 15, but remember for 19, we're only covering, although I'll post the whole chapter for you guys, we're covering that last experiment, Barbara McClintock's experiment 19A, which is in the back of the chapter. Those are the last two things that we're going to cover, and I'll hand you guys the test, right? We'll go over that. I'll bring the other tests back with me too on that Tuesday before Thanksgiving. And then your guys' final is December 1st. What day of the week? I don't even know what that is. It's on a Thursday, right? At whatever time I have posted, we will be in here. Bring your Scantron, right? You don't need anything bigger than the 1 to 100 that we've been using, but you'll probably use to 100.
Will we get a study guide or is it just the objective?
So, yes.
Are we using the Scantron for the test?
Yeah, so for the take-home test, it's still going to be partially Scantron, right? But part of it, you're going to actually write on the test the numbers of the objectives that match the questions, right? So, make it a little challenging for you guys. Not too bad. All right. So I'm going to go back to the office now. I'll post all those outlines and stuff for you guys too. And I'll post that other thing that I got to find that I already did.